Hi, and welcome to our Dairy Innovation Hub research project. This research is being led by Kerri Coon in collaboration with Garret Suen and Johanna Elfenbein at the University of Wisconsin-Madison. I'm Andrew Sommer, a PhD student in Kerri's lab. Our research project seeks to better understand the capacity of flies to transmit disease-causing bacteria to Wisconsin dairy cows. Dairy cows are an integral part of the Wisconsin economy. The state's 1.28 million dairy cows allow farmers to produce over 3 billion pounds of cheese alone each year. However, Wisconsin dairy barns are not without their problems. Cows often share their home with a variety of biting and non-biting flies, which are a nuisance and health concern for the cows, and they may also spread disease-causing organisms. The flies themselves live in close association with potentially pathogenic bacteria. This is because dairy cow manure is the primary site where flies breed and then lay their eggs. Eggs laid by adult female flies hatch into tiny larvae, which feed on the surrounding manure and molt several times into larger stages. When the larva finally reaches maturity, they pupate and eventually emerge as adults. During this time, larvae also ingest bacteria present in the manure, which can persist to the adult stage. These may include pathogenic bacteria like Staphylococcus, Streptococcus, Salmonella, and E. coli. These bacteria are known to cause diseases like mastitis and enteritis in dairy cows. Mastitis is characterized by severe inflammation of the mammary gland and udder tissues, while enteritis refers to inflammation of the intestine. Mastitis and enteritis are two of the most common and costly diseases to the dairy industry and can cause a wide variety of problems for farmers. For example, milk from infected cows often must be discarded, and the infected cows must be medically treated. This results in economic losses of up to $35 billion each year. Our research will examine the presence and abundance of potentially pathogenic bacteria in fly and manure samples collected from dairy farms across southeastern Wisconsin. We will also perform functional assays in the lab to examine the colonization and persistence of known clinical isolates in the housefly Musca domestica. Our results will provide novel insights into the processes underlying environmental persistence and transmission of these bacterial pathogens, and will ultimately help farmers to raise healthier and happier cows in Wisconsin. Thanks so much for watching, and remember, support your local farmers.